Here. Okay. Nice. So it's time for relaxing. Now it's just pretty plots. You do not really run any other code. It's now is so me and Olivia will present VIEWpoly, and now with hidecan integrated. So the VIEWpoly idea is to integrate the results of all the softwares that have been developed, and you run all the codes, and then now it's just time to have fun and explore the results. And so at the beginning of the year, I think January, we had other tools for Polyploid's workshop, and then Olivia spoke there. I was like, another software to include. So now we make this collaboration. We were coding together the last months, and then now hidecan is also inside VIEWpoly.
That's cool. Bravo.
And then I would just present the whole idea of what is VIEWpoly, and then we would just see pretty graphics. So the idea is that because you generated a lot of things, like you have a bunch of linkage groups, you have many results from KTL softwares, sometimes more than one software, because probably we want to try other, like several KTL's software to do your analysis. And you have several traits, you have several groups, you have many KTLs, you have to connect this with genomic information. And also now, because of hidecan, you can also have like differential expression data or candidate genes. And all the combinations of all of these for you to explore your results can be very complicated. And so for this, we create this app, that's a Shiny app, that you can do interactive exploration. And you can easily access all these combinations just changing buttons. And this is good because sometimes, and it happens a lot in our lab, like I produce the results, me and Jikan, we produce the results, but we want to share with David author. And then we just send them the files and then just click this button and you can help me interpret my results. And this can be very like collaborative inside your group. And also, VIEWpoly has a version for servers, so you can just create your own website with your data, and if you want to publish your data to be accessible to everybody, you can do this. And just do, I have a tutorial for that, okay? Huh. I don't know. Okay, but I don't know, but this is the VIEWpoly interface. So we created this using a framework that's very easy to expand. That was how like hidecan was just included. It's just a new tab here. So it's a modular structure that Golang package provides that. So it's easy to expand. And if we are having more softwares that requires visualization in the project, probably I will like, I want to do it. And then I will do the same thing that I did with Olivia and include it here. Okay. And we have many features already. One is just for you input your data, and you have like links for all the tutorials, everything that you need. And once you upload your data, you can visualize each one of these models. One is specifically for KTL analysis, others for the combination of KTLs with the genome, and the other ones for exploring more about your linkage map, and hidecan that I will leave a little bit says later. So this is the input data, and we have specific formats for each one of them. As you saw, like it can ask you to save specific objects, and these will be the inputs for the VIEWpoly. And of course, our data will be easier, but we also have options for if you want to use another software. But this can be a little bit more tricky because you need to put the CSV files in the right format. So it's easier if the software is already implemented. But we have the options to include CSV files too. So I include here links for the writing tutorial, but it's all in this GitHub page. So links for tutorial for everything, and also for how to do with your own data set if you want to deploy this in a server. I will just show here how is this. So it's a single line of code. Okay. So it's VIEWpoly run app.
You said no code.
Just that. And then here it is. Oh, this is the old version. Let me open the new version. This is the version in GitHub. So I updated it. Now I included hidecan here. And the first page has just one about, like just to understand what the software does. And if you are doing a server version for yourself, you can replace this about page with your project description. And I teach how to do that in the tutorial. And this will have like the input data. So each box will be the input files from different softwares. So here's for the package map. So the ones that are available is for VIEWpoly and PolyMapR. And we have one that we just produced here. The one that Genomics asked you to save it. So I can just upload here. And once I do that, I just need to submit. And I will also upload KTL files. We have options. KTL-poly was the one that Jikin showed, but also you can use dia-KTL or poly-KTLR. And if you open KTL-poly, that was the one that we used. We just need to grab the files.
There, first one.
Here. So the data comes here. And here.
Oh, you can drag and drop.
Yes.
I didn't know that.
Emmer figured out that. I didn't know too. Emmer showed me. I said, oh.
Wait, who showed you?
Emmer?
Oh, she showed you?
Yeah. And then this one. And then this one.
This whole time I was.
Yeah. Then I just submit. And then if you don't want to load every time all the files, you can come here and download VIEWpoly dataset, because what these functions do is behind is converting all files to a VIEWpoly format. And at the end, we just keep the information that we will need for the plots. And this object is lighter, so it doesn't have all the things. So you can just choose a name like RGC11. And then you download it. And next time that you use, the only thing that you need to download is this one.
Only thing you upload or input.
And after I can just, sorry? I can just come here and upload the VIEWpoly data. Okay? And because I submitted, if you see here, that's already the groups of all the groups that you evaluate, and this will plot the log profile. You can see only one phenotype, or you can select all of them. And each peak will be, KTL peak will be marked with these triangles, and then the lines are the intervals. And if you want to explore more about each one of the KTLs, you just select it. And then you can have the effect plots. So I can change, like, instead of this, I want to see this, and then plot for you. And then you can download these images in different formats, or you can customize in different ways. For the KTLs, you can also see the effects in different ways, like this is a circle that you can compare the phenotypes. And also if you have like digenic effects and the combination of the effects between the parent haplotypes. Okay. And this was all Jikin's idea about how to, once you have one allele that's important for you, you want to select the individuals that have that haplotype. So you can come here and select the haplotype that has the larger effect. So you have all the haplotypes here. You can just select two of them. I don't know. I will select more. This one and this one. And then you just ask VIEWpoly to search the individuals that have this. And then we print the individuals of the protein that have that specific allele that you selected. And you can also have access to breeding values and a summary of the KTLs. All here. Okay. And the genome, I didn't upload the genome, but I will show you in the here. The genome is a little bit heavy, so I will not upload it, but I can show you from the server version.
Yeah, the Wi-Fi is taking
I will jump to the other tab and then I come back to the genome when it's done. So the map, you can explore aspects of the linkage map. So that plot that you saw that you can show you like with the dosages and the groups. But here you can zoom in and zoom out the places that you want. You do that. You can select a specific place. You can connect this with the KTL peaks. So the KTL's profiles are here. So you can zoom the KTL peak and explore the dosage around there. Here. You can also access the parents haplotypes table. You can download it. And a summary of the linkage map. The main characters like number of markers, size, and gap, and a bunch of things. And the pretty image of the linkage map. And let's see if it's ready. Oh, it's not ready. I will show just the idea here is we have the KTL profile here also that you can go to the peak. And then you can check the relationship between your linkage map and the genome position. So if you have inversions, you can check here. I think Jikin already showed this type of graphic before. This group is very similar with the genome position. We don't have inversions, but if we have, we could check here. Also, we have a JBrowser interface that will not work because I didn't upload the genome here. But let's see. This is, yes. So here works. Yes. So we can relate the KTL peak. This is at end. With if there is inversion or not, and the JBrowser. So if you zoom in here, this is your genome sequence. And also you can include all the tracks that's possible to include in the JBrowser, like the genes and all the things. And you can also download the genes that are located in this region that you selected in this table. Yes, I think I said everything, no?
Yes,
That's good?
Yeah.
I covered everything, yes. So it's your turn. Yeah.
Okay. So as Cris was saying, I've been working on a package called hidecan, which is a package that you can find by itself on CRAN. And basically the idea behind the package is that it's a visualization package where we can see if we have some GWAS results, and we also have differential expression results, so for the same genome, basically. And we also have some genes of interest. So that the reason behind that is during my PhD, I was studying tuber bruising in potatoes. So I had genomic data, so I did a GWAS GWAS analysis. I had transcriptomic data, so I did a differential expression study. And there were already studies about tuber bruising. So I knew already some genes that were found to be associated with tuber bruising. And I wanted to be able to see all of these results into one single plot where I could look at the overlap between the different results. So that's how the hidecan plot came about. So at the beginning, you just have GWAS results, differential expression results, and candidate genes. So again, when I'm talking about candidate genes, what I really mean is any genes that you are interested in. And so the concept behind the hidecan plot is that we're gonna, oh, sorry, it's a bit small. I should have made it a bit bigger. We're gonna combine all of this information into one plot. So that's what a very simple hidecan plot looks like. One box corresponds to one chromosome on your genome. The x-axis is the genomic position. So you have here the beginning of your chromosome all the way to the end of the chromosome. And then you're going to overlap different layers of information or different results on the different tracks that are on the y-axis. So it's a bit small, but we're going to see it bigger later. So it says GWAS peaks, differential expression genes, differentially expressed genes, and candidate genes. So in order to construct a hidecan plot, we have the GWAS results, we select a significance threshold, and all the markers that have a score higher than this significance threshold are the markers of interest. We can pull these markers, check their genomic position, and we plot these markers on the hidecan plot. So you have 1 point for each marker that is above the significance threshold. And just to help compare the different tracks, we have a vertical line that shows us where the markers are positioned. For the differentially expressed genes, we can do exactly the same thing. So when we do a differential expression, typically we want to set a threshold, significance threshold on the score. So all the genes that have a p-value below this are differentially expressed. And also we want to set a threshold on the fold change. So all the genes that have a log2 fold change above 1 are differentially expressed. So that's what you can see here on this plot. We set our 2 significance threshold, p-value or score, and log2 fold change. And that gives us the genes, the interesting genes, the differentially expressed genes. And again, we can take their position and plot it on the hidecan. So now they're going to be there. The position of these differentially expressed genes will be on the second track of the hidecan plot in a different color, and you're going to have again these vertical bars to help compare between the 2 layers of information. And finally, we have our list of manually curated list of curated candidate genes, like any genes that I'm interested in. And we have their position on the genome, so we can again represent them. Now, on the hidecan plots, they will be represented and they will have their name will be displayed, because often you know the gene and you want to see which gene you're looking at. So that's the theory. So the hidecan package allows you to create hidecan plots from GWAS results, differential expression results, and list of candidate genes. Typically, you can either load it as a CSV file where you have, so the big CSV file where you have 1 row per, let's say, marker, and you have the score for the GWAS analysis for all of the markers. Same for the differential expressions. You have 1 row per gene, and then you have the log2 fold change and p-value for the differential expression, etc. But as Cris was showing, sometimes it can be nice if you run your analysis by using a special package that does the GWAS analysis for you. They're going to always format the data in a certain way. So what I've done is that I created some function to grab the output from these packages and convert it in a way that you can make a hidecan plot from it, the same way that Cris was doing for VIEWpoly. And so that's what we're going to see in VIEWpoly. And the other thing is that you don't need, when you're creating a hidecan plot, what I'm showing here is you have the result of 1 GWAS, 1 differential expression analysis, and 1 list of candidate gene. But actually, you can just visualize your GWAS peaks by themselves. It's going to work on the hidecan plot. You're just going to have 1 track, and it's fine. Or say you have the result of a GWAS for 2 different traits. And sometimes if the traits are related, it's really interesting to see whether the peaks in the GWAS are located together or not. So you could also input 2 different GWAS analysis results, and you're going to see them into 2 different tracks. All these kind of things. So that's the theory. Now, as Cris was saying, when I presented my package at the Tools for Polyploid workshop in January, she was really cool and let me put hidecan into VIEWpoly. So now you can just go into VIEWpoly, view all of your linkage maps, etc. And you can also look at your GWAS results and candidate genes, etc. So I think that's, yeah, move on to the practice.
Can I ask a question about the differential? Say you had, your differential expression was between, say, 2 treatments, or like, say, a late or an early cultivar. Is there a way of having that as your differentially expressed data, or is it just up or down?
So the results of your differential expression, the log 2-fold change would tell you, like, if it's positive, it's higher in the treatment.
Okay.
And is that what you're saying?
Yeah.
Yeah. So let's say you have the gene expression for control treatment, you do your differential expression, then you see the genes, you have the list, the results. That's going to be the p-value for each gene. That's telling you whether the gene is differentially expressed between the 2. And then the log 2-fold change tells you about the difference in expression between the 2, like treatment versus control.
Okay.
Is that, yeah? And so then you can, in hidecan, you can visualize the value of the log 2-fold change. So you can see actually these genes are the ones that are more expressed in the treatment. These genes are the ones that are, yeah.
Okay.
Yeah. So in the input data, that's in VIEWpoly. So what Cris was just showing, you can go down. And now there is a place to upload hidecan files. So, so that's either you can, so that's the part where you can input VIEWpoly, where you can input GWAS results. There are 2 ways of inputting GWAS results. Either some file that you saved once you've done the VIEWpoly analysis. So that's what you've done with Jenkins at the end. We run VIEWpoly, then we save the results. So we're going to be able to load that into here. Alternately, you can simply have your result as a CSV file. Now, one thing that is important is that the CSV file needs to contain the results for all of your markers, not only the significant one, but the entire list. The reason for that is that you need to see the entire list of markers to be able to see where is your beginning and end of your chromosomes. You can change your threshold, etc. So we're going to do, so what was the trick? Oh, no, okay.
That's the trick.
That's the trick, okay.
Yes, here.
This one, okay, thank you. And that would be the VIEWpoly, this one.
That one.
Yeah, oops.
Okay.
Oh, no, yeah, because I didn't do it properly. Okay, sorry. Now I will know from
This one would be slightly different than the one they saved. No, it would be the same. It would be the same.
Yeah, it should be the same. So I'll just show you with this one, and then I'll show you also when we put differentially expressed genes, etc. Just so that you can see the different things. So submit. I think we have submitted. Yep. Okay, and now we're going to go and look at hidecan. So I think that's the example data, correct?
No, this is his data. So we'll be
Data that we just ran.
lots of expressions.
Yeah. Okay.
That
Yeah.
shows.
Yeah. Perfect. So in the analysis that we've just run, we've run a GWAS analysis for different traits, and also for each trait we've tried different genetic models, right? And so what the hidecan package will do is that it will create one track for each trait and genetic model. So that's why we're going to see a lot of tracks here. So if we just look at, for example, this first track that shows you the GWAS peaks for the black spot trait with the additive model. Then below that, you have also the GWAS peak for the black spot trait, but for the diplo general model that was used, etc. And then you can see a lot of red line. That's because here you have for the Cercospora, is that how you Cercospora? You have peaks, and you can see that whatever the genetic model that you used, you see GWAS peaks for this trait at this place here. Now the really cool thing is that because the hidecan plot is in VIEWpoly, you have a lot of interactivity. So one thing that you can do is that you can change you can change the thresholds for your significance, etc. Now, in this case, when you run GWASPoly automatically calculates the significance threshold. So when you input the output of GWASPoly into hidecan, it's reading the significance threshold and it sets it for you. So in that case, I don't really have control over that because GWASPoly already calculates the significance threshold. The other thing that you can do is you can select what you want to see on the plot. So here we had a lot of tracks and it's getting a bit messy. So instead I could say, I just want to see, let's say for black spot additive and why not defoliation additive. That. And then now I have a much smaller hidecan plot where I can see all the peaks. Now you're seeing that you don't see all of the chromosomes because by default, in order to make things clearer, if you have a chromosome in which you don't have any GWAS peak, it's not going to be represented. And so you have several options in the hidecan plot, like adding a title, subtitle, and like remove empty chromosome. That's the one I'm mentioning. If we untick that, then you're going to see all of the chromosomes, even the ones that don't have any GWAS peak. But it's pretty boring, right? We don't care about that. So here you go.
So why do some of them have the dots and some of them just the red? The red lines versus the dots.
Oh, yeah, thank you. That's a good question. So every time you have a GWAS peak on one of the track, it's going to create, I'll show that as an example. It's going to create a circle. The color of the circle represents the score of the marker. And then also it's going to make a vertical line that is red, just to say red is a GWAS peak. So then here, for all of these markers, you have the marker and you have the line. And then so this line goes across all of the tracks so that it's easier to compare between tracks. So that's why here you have red and it's coming from these markers. But for this, so for the defoliation additive, you only have a peak here. So that's why you only see points here. The other vertical tracks, they come from vertical lines, they come from these markers. Yeah. I will just, is that okay if I restart that?
Yes, yes.
Just to show the example. So let's say that you didn't use GWASPoly, instead you used another package and you saved your result as a CSV file. Is it on the same folder, the examples? Sorry, should have prepared that before.
Actually, we did a surprise for you now, but you.
Where is it?
Okay.
So I'm going to open them just to show you very quickly. That's okay. So that's what my GWAS results file looks like. So you have one row per marker. These are the IDs of my marker. I don't really care about that at all. What's important is I have information about the chromosome on which the marker is, and I have its position in base pair. And then I have the score, the GWAS score for this particular marker. And similar for, I don't know what to say, similar for the candidate genes, I just have a list with one row per candidate gene of interest, etc. We have, like in the hidecan and VIEWpoly, we have examples. So you can see, like if you want to use it, you can see how to format. So I'm going to load my GWAS results into the CSV. Okay, and also I'm going to load my differential expression results. Oops. And my candidate, I think that's the one. Yeah. And submit. And I go back to check the hidecan. Now you see that this one, now I can move it free. I'll show you in a second. It's just to change the significance threshold for the GWAS, etc. And so we have the new hidecan plot. So here we have the chromosomes are named differently because this is my PhD data. So it's not the example, the data that we've run earlier, it's just an example data set. And we have one track for the GWAS peaks, we have one track for the differentially expressed genes, and one track for my candidate genes of interest. So let's say the interesting chromosome is, for example, chromosome 8, where we have some genes of interest, and we can see some GWAS peaks at the same place. So it's quite interesting because we know we're supposed to see a peak here, and we see a peak here. Then we have places where we have, I'm trying to find a nice one, where we have, like for example, here we have a differentially expressed gene, and here we have a marker. They're not super close, but just as an example. If we had nicer data, it would be, or I mean, if we had really interesting results, we would see them a bit closer. And so what I was, yeah.
So if you, can you, yeah, so you have separated, are those, what are the two boxes, like?
Oh, yeah, thank you. So these boxes are my different chromosomes.
Oh, they're chromosomes.
Yeah, so that's chromosome 0, chromosome 1, chromosome 2, etc. All the way to 12, because this is an example on potato. Yeah, sorry, I should have mentioned that. So like here, I'm looking at chromosome 8. But actually, maybe you should, I wanted to show you chromosome 8, so I can go at the top and see, okay, I only want to see chromosome 8. And then it's coming in a second. Oh, I broke the thing.
Oh, we're toying.
I'll add one more chromosome. Yeah, okay. Oh, yeah.
We need to include
yeah.
reception.
Yes, yeah. Yeah, so we'll work on that. But you can select the chromosomes that you want to look at. So I'll just select them all again, so it looks nice. And one thing is you can, the question that you were asking, so by default, the points that you can see for the differentially expressed genes, their color correspond to the score, differential expression score. So the brighter, it means that the gene is like has a lower p-value. But instead, what you can do is you can, in the options, you can color them by, instead of by score, by log2 fold change. And now, if you have something red, we can see in the legend. If we have something that is very red, it means that it's going to be positive. If it's below 1, so here I'm making a threshold of 1. I'm going to just decrease that to show you the legend. If it's below 1, it means that the gene is downregulated. And so it's going to be shown in blue. It's just coming together. Yeah. So, sorry, if it's below 0, it's going to, like if it's negative, if the log2 fold change is negative, it's going to be, it means that the gene is downregulated and it's going to be represented in blue in the plot. So you can see your downregulated versus upregulated genes. Yeah, and then so here you have options like you can add custom names to your different tracks if you want to say something. So GWAS peak, that's for trait 1. Then you're going to write something and you're going to have something added here. So if you want to customize or add more information. And I think I'm going to stop handling here and give you back.
Yeah, it's just a final slide, just to say that we are still developing. So VIEWpoly is being updated. Now I have this amazing help. But one thing that's it's in our tracks, like include for diploids too, because we don't care about diploids, but maybe it can be useful for diploids too.
We care about highly heterozygous, clonally propagated diploids are always welcome.
Yeah, so we will include for other softwares too, like softwares that do for diploid analysis. So I think for GWAS will be the Lipka, it's how's the name?
Gepit.
Gepit.
Yeah.
And also I can include for ROMAP, that would be easy. And so we can include an ArchiTel. ArchiTel will be a good one. Lots of people use ArchiTel. We'll bring a lot of users. But we will work on that. And also other graphics for showing like the double reduction for polyorigin, something that we will include also, and preferential pairing graphics. Yeah. So, and if you have more ideas, if you have like, I want to see results from the software, just let us know, and then we will include other models, because it's possible to expand.
So one example is like often people use DSeq2 or Lima to make their differential expression results. So one of the things that I want to add to hidecan is the option of grabbing the DSeq2 results and and then with what Cris has done, it's really then easy to you drag and drop your thing and you can visualize directly.
Good. Excellent. Project members, collaborators, and then. Okay, thank you, everyone.
That was a tour de force today.
And I think we have an
It's not over.
It's not over. Excellent.
Something new.
Yeah, okay. It's gonna take 5 minutes, and again, it's just you looking at pretty stuff, or pretty I hope. So.
Yes.
Yeah, well, I set up.
Can you just clarify, to use VIEWpoly, we need the Shiny app?
If you install VIEWpoly, all the dependencies will be installed.
Oh, okay.
And one of them is Shiny.
Okay. But it's within R?
Yes.
Yes.
So you can install the package, like install package VIEWpoly, and the single command that you will need to run is run app. The page for you.
If you get really, really like R savvy, what you can do is you can take her source code, like she was explaining, that very first page where it explains all the introduction and stuff, you can like mess with that and talk about your experiment or whatever, and then attach as a supplemental figure.
Yeah,
Then you can git clone her thing and then freeze a version and then upload your own data so that it is the example data.
Oh, I see.
So that somebody could go explore your QTL data as supplemental data. So that's what I'm about to, the black spots Prospera thing that I talked about. I'm about to submit a paper, and that's gonna be part of my supplementary data. It's like, go to this link. You can look at all the pretty things that you probably.
Yeah, we didn't want to make like the app as a website, as people usually do. So it's an independent package. You can run it locally, so you can test without sharing your data with anyone. But also you can deploy as a website. So if you have a server, it's possible to do that. I wrote a tutorial for doing what Chicken is doing. So it's just there in the VIEWpoly link webpage for making as a website.
Okay.
And if you just want to try VIEWpoly and you don't even want to, like if you're running into issues to install it, you can just, so on the GitHub page, there is a link for a version that is available, like the app is available as a website. Yeah, online. Yeah. Okay. So maybe I'm gonna wait for David to come. But I'm gonna share.
Yes, but VIEWpoly has saved me so much time.
Not yet.
Because for my very first paper, I spent so much time making the figures. And then Cris came along and put VIEWpoly, and then the second paper, I'm just like, oh, this is perfect. You just download the R data, and then if you're familiar with ggplot, and ggpubr, you can download all the things and stitch them together however you want, instead of sitting there like 10 lines of code just for one plot. So, very useful.
Yes, I agree. Okay, so this last section, it's a bit different from what we've heard, but it's just the idea of one more cool thing that you can do when you know a little bit about R to make your life easier by visualizing stuff interactively. So what I want to talk about right now and show you briefly is a tool that I've developed for visualizing breeding values. So the goal was that the breeders have breeding values that are computed, and they want to use that to make decisions for their breeding program. But it can be a bit annoying to look through tables. And so ideally, they would want to have some interactive tool where they can really look into the data and dig through and find the possible parents that would be useful to cross, etc. So I've been using two R packages called Flex Dashboard and Shiny, which is what Cris is using for VIEWpoly, to develop an interactive dashboard where I'm going to show you. So at the moment, this is really just a prototype that I've been working on, but the idea is that once the breeders are happy with what it looks like, we can then deploy it and hopefully even link it to our database. And then we can get the breeding values into the app, and every time someone calculates new breeding values, they're put into the database, and then you can visualize. But at the moment, it's just a prototype that I'm working on my computer. So this is what the main tab of the dashboard looks like. So I'm not going to do live demo. I'm just pre-recording. I've pre-recorded some things to check. But this is what the app looks like. So basically, it's literally a dashboard where you have a side. Yes, that would be better. So you have a sidebar where you have some options where you can control different aspects. And I'm going to go through the different parts in the dashboard. So when you open the dashboard, the first thing that you want to look at is this scatter plot. Now you're going to find that it looks very weird. That's because I just simulated a bunch of random data. So that's why I have random points with random colors and trait names that don't look like anything, because it's just for the demonstration. So this main scatter plot shows the breeding values for different individuals for 2 different traits. So each point represents one individual or one pedigree item, if you want, in your breeding program, for which you have a breeding value. And then the coordinates, so the x-axis shows the breeding value for a certain trait, and the y-axis shows the breeding values for another trait. And so the idea behind that is that you want to select, for example, individuals that are that have good breeding values, high breeding values for the 2 traits at the same time, or something like that. So you can, okay, maybe I'll show you in the demo, and I'll pause when I. So here I can change which trait I'm looking at in the scatter plot by looking at the by going on the sidebar and changing the breeding value of which trait am I looking at. And also the samples can be colored by different things. So just to pause here. For example, I can have a group that could be the species. If you have individual from different species or from different orchards, or I don't know what. One of the examples in my dummy data was if you recorded the pedigree of these individuals, you could color by the female, a parent, for example, or the mother, the father, something like that. Now, starting again, this is an interactive plot. So when you hover, when you put your mouse over points, you're going to see information. Oh, I just stopped at the wrong time. But you're going to see information popping up. So you're going to see it's very small, but you can see the ID of this individual. So here it's just individual 574. But maybe in the breeding program, people will understand, the breeders will understand what they're looking at. And we also show all of the metadata that we have about this individual. So the sex, for example, the group, any group variable that you have, female parent, male parent, etc. And because it's an interactive plot, you can also use the legend to remove points or to just focus on some groups, on some progeny of some parents, etc., by clicking on the legend. Now, the main interest is that we want to look further into some individuals of interest. And so the problem with a scatter plot is that you can only look at breeding value for 2 different traits, right? But actually, often you have more than 2 traits, and you want to select for individuals that are good in all of the traits. And so in order to resolve this issue, what I did is that we have the main scatter plot, but then below that, we have box plots that show the distribution for additional breeding values for other traits. When I'm going to select, so this scatter plot at the top is interactive. So when I'm going to select individuals that I'm interested in, so here I'm picking individuals in the top quadrant of the plot, so they have high breeding values for both traits. When I select them with my mouse, I'm going to see here that the points, like it highlights these same individuals in the other box plots. So I can see for example, the breeding value of these individuals for other traits. And so again, these points, if I hover my mouse over them, then I can see information. And here on the sidebar, I can select which traits am I seeing in the box plots. So I can add more or remove them, etc. Now, the other thing is the last part of this main dashboard tab is that you have some tables on the side. There are two different tables. The one that you see when you open the app is just information about the individuals. So in here, I call my individual pedigree item, but it's just an individual. And you have some metadata about them. So the sex, the group, the parents, anything that you want. And so by default, it's going to show. So by default, it's going to show all of the entries that I have in my data set. So if I'm unclicking, so I'm not selecting anything, you see that I have 600 entries in my dataset. But when I select points in my scatter plot, then I see only information about these individuals. So that's the first tab. The second tab is just the breeding values for the different traits. So the actual values that are shown in the scatter plot and the box plot. Now, the interest of this table is that they are interactive as well. So I can search. So I can search for an individual and see the breeding values. Or I can order. So I can reorder my table so that I can see the individual with the highest value for a certain breeding value for a certain traits. And I can also download. So sometimes what the breeder says is once they've had a look, they want to have a table and they can go through it in more detail. So you can click on the table and download the table, either the entire table or just the ones that you selected. Yes.
Go back. Can you, if you hover on the right, will the, can you get it to talk to the plot lead to have it show up?
I could. I haven't done that, but I could do that. Yeah.
That would be interesting.
Yes. Yeah.
As a breeder.
Yeah. Okay, that's cool. Because so the thing is, I started developing this app. I just had I was told make a interactive dashboard. So I do something. But then oftentimes, I don't know what the breeders want. So then it's really nice to have input, and then I know what I can change. So yes.
Can the plot lead do lasso?
Yes, I think you can select here.
Okay.
Oh,
so that it doesn't have to be rectangular. I think it's that one app. I think it's right.
Yeah, so my mouse is not on the same.
I think I've seen it before.
Here,
yeah,
like here, this one.
Yeah.
Which is, it's not at all on the same place. Yeah, so definitely it can do that. So that's really
that would just be really cool, like
make
everything else gray and then have that one pop up, or maybe some kind of big red circle so you see where it is.
Yeah, definitely. And the other thing that you can do is you can zoom onto the plot. So like you can focus on a certain distribution of breeding values or something. Now, when I showed that to the breeders that were interested in this dashboard, their question was, well, that's nice, but usually we don't look at individual breeding values to make a decision, but rather we use a selection index where we're going to weight different traits and we're going to assign a higher weight to traits that we're more interested in or something. So that's this one. So there is a tab, the second tab. So this is, again, I don't know why it's not, okay. Here, that's the main tab, but the second tab is something about creating the selection indices. There are two ways to do that. If you are just exploring your data, you can create selection indices individually here, and I have a little film to show you how to do that. The other option is to upload a matrix of breeding value selection indices. So let me show you the little movie. So I'm in the main tab, and I'm just going to go and click on the second tab. And I'm going to start by creating a selection index here. So the first thing you do is saying how many traits do you want to contribute to your selection index. 1 second, it's coming. Yeah, here. So here I'm saying, okay, I want to create a selection index with 3 different traits. I can give my selection index a name. So I don't remember what I typed. I want color. Okay, why not? And then I'm going to assign weights to my different traits. So either you can, by default, by clicking on the arrows, you select integer weights. They can be positive or negative. or you can put a value between 0 or 1 and 1 or whatever. So I think I'm showing here, 2.5. And then in the drop-down menu, I'm going to just pause that so it's in the drop-down menu, you select which trait you want to use. So here I'm creating a selection index that's using traits. Let me just do that quickly. That's using trait D with a weight of 1, trait E with a weight of 2.5, so it's going to contribute more. And I don't want to select for trait F, let's say, so I put a negative weight. When I press on generate, I'm going to see at the bottom a summary of what I've created. So I've created an index that's called I want color, and I have a trait of 1, for, weight of 1 for trait D, weight of 2.5 for trait E, etc. Just showing that. Now, I can start again. So let's say I want to create a second index. Just showing again. I can put whatever values I want. And that's the reason why I created 2 in the little demo, is just to show you, as you create more, you're going to see only the interesting trait that pop up at the bottom. Now, once you've created your selection index, you can go back to the main tab. And now on the scatter plot and on the box plot, you have this selection index that you can select to visualize stuff. So by default, it's going to be added as the x-axis of your scatter plot and as a box plot. And when you go to your table of breeding values, it's also, there's going to be a column that is created to visualize, like to look at the values for that. Now, the other way of creating a, let me stop that. The other way of creating a selection index is using a template. And that's because once you've created your I want color, and every time you want to use it, you don't want to every time click, okay, I want 3 traits, and remember the weights, etc. So instead, you just want to upload a matrix that has all of your indices. So in order to do that, there is a link that I just clicked on in the demo here, and you download the template. So it's just a CSV file where you have the first column is for the name of the index, and then you have 1 column for each trait that is present in the data set, and you can pick weights. So here I'm going to change the name, maybe. Yeah. So I'm creating a new index, and then I'm picking different weights for the different traits. The advantage of doing that is that you can create different selection indices at once on the different rows, and you have it every time you want to restart a new visualization, like a look at it again, then you can use it. So once you've created your template, now you can upload it into the app. And so you create your template once, and the next time you just have to upload it. So when I upload it, automatically I've seen that I've created a new index. So that's my new index. And if I go again on the main tab, then my new index has been created. Once I'm done, I can then download the table of all the selection indices that I've created. The reason for that is the first time you play with your selection indices and you create them and you're happy with them, you can just download and next time upload that. And so that's what it looks like. A table where you have a column for the name of your index and then the weight for the different traits.
You've got a question on your breeding values that you're going to use for the index. Are they deviations from an intercept, or are they in the unit of the trait?
They are Z-score transformed. So they, That was one of my first mistakes. I was really happy. I showed that to the breeder, and what's the unit? And so they wanted a Z-score transformation.
Exactly. That's what we use.
Yeah, awesome. The last thing, so there's no little clip for that because it's just boring table. But more seriously, so one of the things that the breeders gave me as a feedback is that it's also nice to be able to look at the raw data. So the breeding values, you calculate them by fitting a model, but it's model that is created based on the results are created from raw data. So that it would be good to be able to see the raw data. So the third tab of the app is just basically the table of the raw data. So you have the pedigree item or your different individuals, and then let's say that you evaluated them during different trials across different years, and then you have the raw values for the traits that are measured. So that's just your table. Again, it's searchable. You can also filter based on certain values or certain years, or you can reorder, etc. And by default, you see all of the data. But in the main scatter plot, if you select your points, then you only see the raw values for these individuals that show up in the table. And then you can, again, download if you want to look at it further. So yeah, just as a bit of a conclusion. So as I was saying, I'm working on creating the app, and once the breeders are happy, then we would like to deploy it to make it available through the breeding database. And also currently, the way I'm building the interactive dashboard is by reading the breeding values from a CSV file. But actually, in the future, it would be linked to the database. So I read the breeding values directly from the database.
What platform do you use for that?
Suzanne. What platform do we use for the database, the breeding database? That would be
Bracket?
no. Why did I forget the name?
Honestly, Charlie, but I'm not sure.
Yeah. That's for our Kiwi Cloud
We've got
database.
two
Yeah.
databases, and I can't remember
Yeah.
what you breed it as either.
Yeah.
Yeah, we should.
It's a work in progress.
Sorry.
Yeah.
Be integrated into Bracky, and a lot of US people use it probably.
Yeah.
Yeah, they are doing a bit of work in trying to get some Bracky tools installed. Yeah, there's a lot going on there.
Yeah.
But even from static CSV, this is amazing.
Yeah. And so, yeah, that was just a bit of a visualization showcase of the kind of things that you can do with things like a shiny flex dashboard that can really make the life of breeders easier without having them to go through the code or go through Excel files, etc. So,
That would have made my talk in
All right.
I had this, that'd be amazing.
yeah, and it's not really complicated.
Keep waiting. Yeah, bust it out.
Yes.
Oh, yeah.
You got here about 20 else. Probably wanting to move.
So we can talk about the
Thank you.
developing on an action platform.